SCRaMbLE and its use in synthetic biology. In synthetic biology, DNA is engineered to rebuild pathways and constructs in order to produce something new and different. As the concept of synthetic biology has grown within the scientific community, tools and techniques to engineer DNA have been developed. Such techniques include the CRISPR-Cas system, or clustered regularly interspaced short palindromic repeats, as well as Gibson assembly. Another technique being used in synthetic biology enabling the rapid and effective engineering of DNA is SCRaMbLE. SCRaMbLE stands for Synthetic Chromosome Recombination and Modification by LOXP-Mediated Evolution. In 2011, a consortium of scientists working on a project called Yeast 2.0 first described the use of the SCRaMbLE technique to partially assemble a eukaryotic chromosome. It was demonstrated that the SCRaMbLE technique produces complex genotypes and phenotypes which are different to the native ones. In 2014, the technique was further used by the same group to assemble the first ever synthetic eukaryotic chromosome. They called the synthetic yeast chromosome SYN3. This was a big step forward in the Yeast 2.0 project and in synthetic biology. Further validation of the SCRaMbLE technique was presented in 2016 when SCRaMbLE's ability to produce large amounts of genetic diversity and identify fitness constraints were examined. So how does SCRaMbLE actually work? SCRaMbLE has been described as an inducible evolution system and is based off the Cre recombinase and LOXP system, generating large amounts of DNA through translocation, inversion, or deletion. The understanding of the Cre recombinase and LOXP system arose when LOXP sites in bacteriophage P1 were observed to undergo site-specific recombination. In 1984, this understanding was further developed when the Cre recombinase enzyme was observed to be interacting with the LOXP sites. In 1987, Brian Sayer demonstrated the bacterial width system within yeast, and in 1992, the Cre-LOXP recombination system was demonstrated in transgenic mice, where it was observed that the LOXP flank DNA could be deleted from genetic sequences. These initial developments led to the use of the Cre-LOXP system across many different organisms, including in neuroscience, cancer studies, and the assembly of artificial chromosomes. The Cre-LOXP recombination system involves the use of sequences called LOXP sites and the Cre recombinase enzyme. Both of these parts of the system come from the P1 bacteriophage. The recombination event occurs when the target piece of DNA is either inverted, deleted, or translocated. The recombination event will only occur when two LOXP sites, each 34 base pairs in length, are present either side of a target piece of DNA. The 38 kilodalton Cre recombinase enzyme recognizes the direction and positions of the two LOXP sites, catalyzing the recombination event. The direction of the LOXP sites indicates to the Cre recombinase in what manner the recombination is to occur. The advantageous feature of this system is that for recombination to occur, both the LOXP sites and the Cre recombinase enzyme must be present. Additionally, due to the short nature of the LOXP sites, homologous recombination should not occur. The designers of the SCRaMbLE system wanted to remove all non-essential genes from the genetic material in order to build a minimalistic, functional eukaryotic chromosome containing only essential genes for the chromosome to work. To do this, they altered the LOX P sites to be symmetrical in nature and positioned them in the 3' untranslated region of non-essential genes. These sites were called LOX P sim sites. Due to their symmetrical nature, they were able to facilitate recombination events in either direction, increasing the number of rearrangements that could be generated. Their design enabled them to generate large amounts of recombined DNA that was very different to any of the native pieces that they began with. The team also modified the Cre recombinase enzyme to contain an estrogen binding domain, allowing it to be inducible and its presence controlled. Since its initial presentation in the literature, the SCRaMbLE system has been applied to the yeast system throughout the Yeast 2.0 project, with the first ever functional eukaryotic chromosome produced in 2014 based off the system. More recently, adaptations to the SCRaMbLE system have been reported. A demonstration of the system being used in vitro was presented in the literature this year, potentially allowing for more control and knowledge over what exactly had been scrambled. Additionally, it has been reported that a live control Cre-mediated recombination system based off SCRaMbLE provides greater amounts of control over the recombination method. The SCRaMbLE system enabled the modification and assembly of synthetic chromosomes. Continuation of the use of such a system could lead to the assembly of all the chromosomes of a synthetic organism. The tool is an appropriate one to use when trying to generate minimalistic genomes. However, at the moment, its uses in other systems are limited.